I now call to order the Society's 2,456th meeting in what is now the 151st year since its founding on March 13th, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends to tonight's lecture by Kimberly Prather. Before we begin, I want to note that it's Women's History Month. And the Cosmos Club and PSW have a long relationship. The club was originally founded by mostly members of PSW, and PSW has been meeting continuously in the Cosmos Club since the Cosmos Club first had a permanent place to meet. However, the Cosmos Club took a very long time to recognize the power, creativity, and capacity of women. And it wasn't until 1988 that it had its first class of women members. And among that class were Susan Tolchin, Anne McLaughlin, Esther Peterson, Helen Hayes, and our very own Rita Caldwell, who is on our call tonight. Hello, Rita, and congratulations on breaking that barrier. We're very glad that you are on the call tonight. Tonight's meeting is being held via Zoom so that our speaker, Kim Prather, can join us from her location in La Jolla, California. As of now, we are planning to have the next meeting in person, but this will depend on COVID and the measures instituted by government authorities and the Cosmos Club. The venue will be posted to the website and highlighted in the announcements that will be sent before the lecture to the extent possible. But please check frequently for updates. All in-person meetings, of course, will adhere to the Cosmos-related rules of the District of Columbia and the Cosmos Club. We will be live streaming all meetings to YouTube for our remote audiences to enjoy and to ask questions on the YouTube chat box. And final versions of the recordings will be posted to the PSW website and to the PSW YouTube and Vimeo channels, where they will be permanently available without charge for everyone and anyone to view. The Society is grateful to the sponsors of the 2021-2022 lecture series for their support. We note the Policy Studies Organization in cooperation with the American Public University, and also a generous donor who has asked to remain anonymous. And we are also thankful to the sponsor of tonight's lecture, who also asked to remain anonymous. Please join me in thanking all of our sponsors. Thank you, sponsors. I am pleased to announce the following new members. Herbert Gintis, an economist with the Santa Fe Institute, interested in sociobiology, who learned at PSW from the internet. Michael Wilson, a computer scientist and software engineer interested in quantum computing, programming languages, computational ecology, and conservation biology, who learned of PSW from Eventbrite. Jean Morrow, a physician interested in chemistry, biology, biomedicine, and health sciences, who learned of PSW from PSW corresponding secretary, Robin Taylor. And tonight's speaker, Kim Prather, who learned of PSW from our invitation to her to speak tonight, and whose interest will be clear in part from tonight's lecture. We welcome them all to membership. If you're not a member and would like to join PSW or support the Society, you can do so through the PSW website using the blue Join button on the upper right-hand corner of the homepage. We welcome new members and appreciate donations. Recording Secretary James Heelan will now read the minutes of the 2,456th meeting and the lecture by Melissa Hawkins on squirrels, lemurs, and all manner of creatures, great and small. James.
Good evening, everyone. On March 11th, 2022, from the Powell Auditorium of the Cosmos Club in Washington, D.C., and by Zoom webinar broadcast on the PSW Science YouTube channel, President Larry Milstein called the 2,455th meeting of the Society to order at 8:04 p.m. Eastern Standard Time. He welcomed new members, and the minutes of the previous meeting were read and approved. President Milstein then introduced the speaker for the evening, Melissa T.R. Hawkins, curator of mammals and research zoologist at the Smithsonian Institution's National Museum of Natural History. Her lecture was titled Squirrels, Lemurs, and All Manners of Creatures Great and Small, Field Studies and Museum Collections, Essential Tools of Taxonomy, Ecology, Evolutionary Biology, and Conservation. Hawkins is principally a mammologist interested in evolutionary biology or systematics, specifically in adaptive radiations in Southeast Asia and Madagascar. Both geographic regions have experienced significant deforestation since the mid-20th century. Hawkins uses museomics, the large-scale analysis of the DNA content of museum collections, to analyze the mammal collection at the Museum of Natural History, the world's largest. The depth and breadth of the collection allows Hawkins to conduct research on remote populations without needing to travel the globe, and to sample extinct animals without a time machine. Her research relies on collecting what she calls crusties that survived museum sterilization processes. Less than 3% of the museum's mammal collection has tissue to sample. When Hawkins is able to extract a DNA sample, she tries to recover signals for reconstructing phylogenies and look at population genetics. She described her research methodology and instruments, then described her museomics research. That work has resulted in published papers on looking at recovering a wide variety of mammalian mitochondrial genomes and the ectobates array. The speaker then turned to her field work. She first described her work studying the colonization patterns of squirrels in Sulawesi, an island comprised of 5 fragments of land masses east of Borneo. Sulawesi has 10 named species of squirrels, adapted into 3 genera. In the last 20 million years, Sulawesi has fluctuated between being entirely above water and having only certain incongruous peaks and ridges above sea level. Based on museum specimens, Hawkins and her team were able to construct an adaptive radiation from the whole mitochondrial genome of about 16,500 base pairs of DNA. After initially believing she identified a new genus of squirrel, she learned her data actually showed a mitochondrial capture from an unidentified or extinct squirrel species. Her results indicated that various squirrels were isolated during periods of high sea level and speciated when they were able to interact as water levels dropped. Given the opportunity to return, Hawkins would have finer scale sampling and do a whole genome sequencing for each genus to look at when they split from one another. Hawkins next discussed her research on the genetic structure of the mountain tree shrew in Borneo. These mammals have an unusually large elevational range, found in every sample from 836 to 3,356 meters above sea level. The research team used UCEs and mitochondrial genomes and determined there were 2 divergent haplogroups with significant geographic overlap. Hawkins said the tree shrew evolved on the island's older mountain, where the data showed greater genetic diversity, and rode the newer mountain as it grew. The data showed the tree shrew had surprisingly complex migration patterns, meaning that despite steep elevational gradient of the 2 mountains, substantial gene flow occurs for the species. In the future, Hawkins would like to expand sampling to the full extent of the tree shrew's range and do microbiome research. The speaker then talked about her research in Madagascar, an island of diverse microclimates that experienced 4 independent migrations of mammals: lemurs, tenrecs, rodents, and carnivorans. Hawkins was tasked to develop the largest phylogeny of Shafaka, the 2nd largest lemurs, weighing up to 6.5 kilograms. There are currently 9 named species. Based on 200 samples, the speaker's team identified 2 clades in the diademid Shafak, which almost perfectly corresponds to the current climate zones in Madagascar. Demographic reconstruction supported the same result. An analysis of 20 years of morphological measurements also showed the clades pulling apart. Hawkins intends to take samples from the British Museum for historical data points to determine whether her research can conclusively establish 2 species of diamond shafak. The speaker also discussed her research on the greater bamboo lemur, also in Madagascar. Genomic analysis showed dramatic historical population increase and collapse. The speaker then highlighted her in-progress research projects and took questions from the in-person and online viewing audiences. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. President Milstein adjourned the meeting at 9:49 p.m. The temperature in Washington, D.C., 11 degrees Celsius. The weather, partly cloudy. And the number of in-person attendees and concurrent online viewers, 62. And number of views on the PSW Science YouTube and Vimeo channels in the first 2 weeks of the lecture, 141. Respectfully submitted, James Heelan, Recording Secretary.
Thank you, James. Send any comments on the minutes to Corresponding Secretary Robin Taylor at corresponding@pswscience.org. That's corresponding sec@pswscience.org. The minutes will be posted to the website in due course, corrected or revised as advisable in light of any comments that are submitted. We now turn to tonight's speaker, Kim Prather, and her lecture on ocean microbes, aerosols, climate, and health, tiny critters, huge impacts. Kim is Distinguished Professor and Chair in Atmospheric Chemistry in the Department of Chemistry and Biochemistry at the Scripps Institution of Oceanography and the University of California, San Diego, where she is also the founder and director of the SIO-UCSD NSF Center for Aerosol Impacts on Chemistry of the Environment. Her research group performs worldwide studies of atmospheric aerosols with a major focus on how they affect clouds. Kim's research has shown that aerosols travel very long distances and affect environments very far from their places of origin. She has shown, for instance, that dust and microbes from Africa can seed clouds and enhance snowfall over the Sierra Nevada mountains in California. In addition to her fieldwork, she and her colleagues have recreated the ocean atmosphere system in the laboratory on a large enough scale to empirically study how ocean phytoplankton, bacteria, and viruses can influence atmospheric chemistry, clouds, and climate. Recently, Kim has studied the role of aerosol transmission in the global spread of COVID-19, and she's advocated widely for aerosol control to help end the pandemic, something you'll hear about tonight. She's an author on over 230 publications, and she speaks frequently at technical conferences and to the media. Among many other honors and awards, she's been awarded the AAR Whitby Award, the ACS Field and Franklin Award for Outstanding Achievement in Mass Spectrometry, the Pittsburgh Analytical Chemistry Award, and the Hagen-Smit Clean Air Award. She is a member of the American Academy of Arts and Sciences, the National Academy of Engineering, and the National Academy of Sciences, and she is a AAAS Fellow and an AGU Fellow. She earned a BS and a PhD in chemistry at the University of California, Davis. All questions will be fielded after the lecture during a Q&A session. Kim, the screen is yours.
Okay, well, first of all, I'd like to thank you for the kind invitation to speak tonight, and also for that wonderful introduction. I'm very sorry that I can't be there in person. I hope I'll be able to be in Washington very soon. I miss travel, but still it was right on the edge, and I'll talk about that a little bit tonight. So tonight I'm going to sort of cover a pretty broad range of topics. And, basically all has a major center around airborne and also oceanborne or waterborne microbes. And I've had a longstanding interest my whole career, 30 years now. I've focused on aerosols. And the funny thing is, people didn't really know what aerosols were until the pandemic. A lot of people thought of the stuff that comes out of a spray can. Now people know what aerosols are. And so, I want to talk about sort of how I'll start with like my main research. And then I'll finish with sort of how did I get pulled into dealing with trying to be a major advocate or an activist. I've been called an aerosol activist in the past. So I'll sort of give you the whole story, and hopefully everything will tie together at the end. Okay, so aerosols. So, this is where my mind was when I was first asked, do I think aerosols can go further than 6 feet? So I study, as was mentioned in the intro, I study aerosols and how they move all the way around the globe. And it takes about 2 weeks. They can get kicked up into the jet stream and they move around. And so these different colors are just things like dust, sea spray, wildfire smoke. These are the common, air pollution. These things can get around all the way around, and we share, basically we share these. And so it's really truly a global problem. And my interest has been how in the center that I direct, we look at how these aerosols seed clouds. So aerosols, basically without aerosols, you don't have clouds. At the center of every cloud drop, at the center of every ice crystal is an aerosol particle. And different particles of different compositions from different places can or cannot make a cloud. And so we're really interested in how chemistry affects cloud formation. And so they span a wide range of sizes, all the way from a nanometer up to about 100 microns. We don't talk about things much bigger than 100 microns too often because they're so big, gravity, they just fall to the ground very quickly. In fact, most of our research focuses on sizes below a few microns. Those are the things that can actually stay up in the air for days to weeks. And so you can see they can get incorporated into storms. They affect intensity of hurricanes. They just play a major role in affecting not only where clouds form, but where our water falls, as I will talk about to kick this off. So as we know, as our climate is changing, we're seeing a big change in extreme weather events. We're seeing evidence, basically there's like, things have moved from, sort of an average amount of rainfall kind of everywhere to we're starting to see more evidence of flooding, intense flooding in some places and drought in others. And one of our, part of what we're thinking is that a lot of that has to do with the aerosols that are actually seeding those clouds. So how could aerosols change where the water actually falls? So I got involved in this first in 2009 is when I started having conversations with the state of California. The California Energy Commission came to me and said, you've developed this instrument, which I'll talk about in a second, but we can actually fly through the cloud, sample the cloud residues, what the cloud drops and ice crystals are, and say where they came from. And we can actually say, basically determine whether it matters what seeded the cloud, or if the aerosols really are changing things or not. And the thinking at the time was that the air pollution in California was just going up in the clouds. It was creating too many seeds so that basically each droplet was basically remaining tiny and basically too tiny to fall. So we were shutting off precipitation. That's what we went into this with. So this is the plane that we flew on. This is the DOE G1. This is me telling the pilots where to fly as we could look at the data coming in. We sample the aerosols, we sample the clouds through these different inlets. There's like at least 50 instruments on this aircraft giving us data of all different sorts, but these are the main ones that I'm going to talk about tonight. Then there's these little things hanging on the wings that basically image the clouds. They tell you if there's ice in the cloud, they tell you if there's water, how many droplets, the size of the droplets, they tell you all the microphysics of that cloud. And I have to say, this was quite a controversial project. This is when I learned, like, the more pushback you have, the harder you should keep going, because basically in meteorology at the time, they were like, who cares what seeds the clouds? If it's going to rain, it's going to rain. If it's going to snow, it's going to snow. I mean, it doesn't really matter. And so we just went in like open-minded, like, let's just see if the aerosols matter or not. So this is the aircraft instrument of our ATOF-MS, which I'll show how it works in a second. This is all the students that worked on various aspects of the project at various times. Carrie Pratt, who's now a professor at the University of Michigan, Dr. Jack Cahill, Caitlin Suski, and Jesse Creaming. So these are all Prather group members, and they've all been involved in various aspects of flying through clouds in my group. So this is how my career started, was instrument development. There was no instrument to measure aerosols, especially single particles. So aerosols are just solid or liquid droplets or, pieces of specks of dust that are suspended in the air. That's the definition of an aerosol. But what people typically had done was just to sample all of the aerosol on a piece of filter paper and then analyze it chemically later, like sometimes months later. So there was no way to really measure an aerosol, the particles themselves, and know what you were breathing right now. So that was the whole goal when I started my research, as I say, 30 years ago. So this is an online single particle mass spectrometer that we invented. And basically it sucks the aerosols in. It tracks them between lasers. So it measures how fast the particle is moving. That's how we get the size, which is really important from a climate and a health perspective. And then we know where the position of that particle, and then we blow it apart with a laser and we get the chemical composition. And we do that very fast. We can do up to 50 per second. I think now with technology changes, we can go even much faster than that if we need to. But the idea is instead of just getting sort of an average composition and assuming every particle looks the same, we get very discrete compositions that allow us to say where those particles come from. And so we can just count up the particles and know, the sources of the particles in the atmosphere immediately. I'm not going to go into this in too much detail. I just want to give you an example of what the data, like the single particle mass spectra, look like. These are the typical spectra for bioparticles, which I'll talk about quite a bit tonight. Biomass burning, dust that comes from all places, Africa, China, very different, different deserts have different compositions. And then dry lake bed salts, we published some papers on showing how profoundly these can actually change the clouds and the climate. But my point here is that, if you just collapse these signals all together and just average them and got it, you wouldn't have a clue, what is, whether aerosols are affecting clouds or climate or not. And so, basically this has been a really big breakthrough. And when we first did this study, we were the very first. They've been flying through clouds looking at the physics for a very long time, but nobody had ever looked at the composition. Nobody had ever said, is there a link with the aerosol sources or not? So we did that. And we had a big surprise. And this is probably one of my biggest sort of claims to fame. It was a surprise to me too. And it took us several years to get this paper published because people just couldn't believe it, quite honestly. But we basically found that it wasn't the local, and sometimes it's the local pollution, but what had the most profound effect on the precipitation in California was aerosols that came all the way across the Pacific from, over 10,000 miles away. And what happens is those, dust and biological aerosols make the magic ingredient in clouds that leads to the most efficient precipitation, which is ice. If you don't have these particular types of aerosols, these sources, the water, they'll just stay liquid. They can stay super cold liquid to negative 38 degrees C. But with bacteria, you can form ice at temperatures as warm as negative 1 degree C. Dust isn't quite as good. It's more like negative 15. But the point is, once you make ice in the upper layer of the cloud, Mother Nature figured out how to get the most water to fall. And what happens is that ice forms, it steals the water from everything else inside of that cloud, grows these, and they fall, and they scavenge everything. So a huge fraction of the water falls to the ground, in this case as snow. And so this leads to a tremendous enhancement in snow. And we've been doing cloud seeding in California since the 40s, but we haven't been doing it very well. Based on how this works and this mechanism works, the natural mechanism works, we actually would be offsetting it. And I'm happy to talk about this more later, but I just wanted to sort of set the stage with how important airborne microbes are in terms of, water, climate, everything. And this was my first full appreciation, just the beginning of my love for microbes in the atmosphere. So it made the news in a big way. People were like surprised. We found these microbes. They were, are they alive? Where did they come from? And so, it was pretty exciting time for us. As I say, it was a lot of pushback. So then the question became, where do they come from? We know a lot more about microbes in soil, terrestrial microbes. It's just easier to go collect those than it is the ocean microbes. And so a big push for us was to really start to understand the ocean. The ocean is a natural source of microbes and gases and everything else that changes the composition of the atmosphere. The ocean covers 71% of the Earth, yet we know very little about what it's doing to our planet, to be honest with you. It's just such a massive portion of our Earth. It's shocking that we don't know more than we do. but it's a really hard problem. So that leads me to, that's what I convinced the National Science Foundation of this very issue. I had done field studies for 20 years. We'd developed all these great instruments. We'd go make measurements in the field and things would change, but we never could really explain why. We would come up with a pretty good story of why, but we were never 100% sure, because in the real world, 20 things can change on you at the same time. You have no control. So you just take the cards that are dealt to you and you make the best story you can. So I was not satisfied with this as a chemist. And so me and a number of colleagues, Vicki Grassian is the co-director, Tim Bertram is the associate director, and a whole lot of other people got together and basically proposed that we would look at how aerosols affect climate, but we would do it in a unique way. We would do it as chemists would do it. We would do the reactions in the lab, but we would use the real system. We would move the entire ocean atmosphere into the lab where we could control the water, the air. We could basically, the idea is we would control everything. And then we could perturb it in some way and then look at what's coming out and how that is potentially affecting our climate, with the ultimate goal to be to improve climate models, which have a very difficult, and atmospheric chemistry models for that matter, they have a very difficult time at dealing with these aerosols. So think about, you step back, what had been done in the past to simulate what comes out of the ocean. So one of the main things the ocean does is it produces a lot of spray. You see that. And people assumed that the best proxy for that is sodium chloride. That's what's mostly in the ocean. And so if you think about this, one of the most profound ways that the ocean changes the composition of the atmosphere is by the reactions that occur on the surface of those particles, the heterogeneous reactions. It changes the entire composition of the atmosphere. But if it isn't just sodium chloride, if there's something else that's coming out, we can study the sodium chloride and learn fundamental things which are really important. But our goal was to really be able to do these studies and be able to explain the real world. So we have to step back and think about the ocean as it is, which is a living environment, produces over half of the oxygen we breathe. It has every component of life in it. And so what happens is a lot of these things don't really want to be in water. They don't love water. And so they will tend to float and kind of sit on the surface of the ocean in this thin skin. And so there's just this incredible world of biology that sits on top. It's also still in the main water, but it's definitely enriched in this, what we call the sea surface microlayer that sits on the very top. And so it's like a forest underwater. It's what, basically it emits gases. It does all the things a forest does, but that's not how it has been viewed over time. So if we think about phytoplankton blooms, again, it's a forest. The plants are the phytoplankton. And one of the hypotheses decades ago, it's called the claw hypothesis. And that hypothesis said, if things start to get too warm, that basically maybe the microbes could emit different things, change the properties of the aerosols, which could change the clouds, how bright they are, how reflective they are. And that could end up cooling our planet. So maybe there's this natural thermostat, just like we have in our bodies, that microbes are constantly trying to keep our bodies in check. Maybe they're also trying to keep the earth and the temperature of the earth in that nice, narrow, happy range. And so, this was sort of the basic hypothesis or idea that we set out to look at in a more controlled way in the lab. So it's not easy to do this. We're chemists and we were trying the best we could, but the beauty of a center is you can bring in experts in every field. So we brought in microbiologists who helped us figure out how to do phytoplankton blooms in controlled ways and to produce real conditions, not crazy conditions, but like really simulate what happens in the real world. We consider these microbes, at the time, we just basically call them our organic chemists. They're organic synthesizers. They give us that complex array of, they estimate there's up to 10 to the 15 different organic molecules in the ocean. And so then we worked with oceanographers who taught us how to look at the transfer. It is not as simple as just sticking straight bubbles. There's a very characteristic bubble size distribution. I've grown to love and appreciate bubbles more than I ever thought was possible. But if you don't get the bubbles right, you will transfer the wrong things, or you won't transfer anything, or you'll transfer everything. But the point is, you'll get something in the atmosphere. But again, our goal is to get the right stuff into the atmosphere, the stuff that looks just like the real world. Then we can actually do reactions finally, that's our starting material. So as chemists, we're happy at this point. We can push it a little bit further and think about how that surface, that interface affects water uptake. So again, cloud condensation nuclei, ice nucleation, how do those things, behave in the atmosphere? And one other thing that sort of we didn't say, and I have to say it for the sake of the story tonight, is that we didn't think about the gases. We just thought it was the spray. I think most people thought it was the spray. But it turns out microbes are constantly, as we know from terrestrial sources, they're constantly emitting gases. But they've been ignored, largely ignored. Everybody's favorite gas is dimethyl sulfide, but there's a whole bunch more. And that kind of makes sense. But why did we not know this? They're incredibly reactive. So in the real world, if you're not sitting in a ship at the right place at the right time, they're there and they're gone in seconds, minutes. And so in our lab, we have a clean head space, so we can pull them out and then react them, as I'll talk about today. The other thing I want to mention really quickly is we went, just as a little side point, because I'm not going to talk too much about this, we went into basically a huge amount of effort in trying to sort of think about what a single sea spray particle looks like. And if you look at the model system, it was just this salt with this beautiful surfactant coating that would block reactions. And that's not what we were seeing in the lab. So now we're working with Romy Amaro and her group, and they've been simulating these crazy complex systems. And they show that actually you don't get that beautiful order. And this explains, we can use this to actually explain what we're seeing. But the take home here is that, was it worth it? At least from a chemistry perspective, was it worth it to go to all this effort, 10 years, 11 years now to do this? And, basically, I think the answer is yes. And so this is one of the systems we use to generate, it's a wave channel. So it's 32 meters long. And I want to make sure is this blocking? I'm worried that this is blocking you. I don't know. Anyway, I'm going to put it off to the side. Okay, sorry about that. So we can basically take real seawater from the ocean, and we can add nutrients, just like dust depositing to the ocean, and create this bloom. And what you see is this is fluorescence, which is indicative of the amount of biology that's happening. It starts to change in the water. And the question is, what gets into the air? And so this is like kind of a quick snapshot of a lot of work where we actually were able to show there was a huge debate, and there still kind of is, whether the biology actually changes the chemistry enough to change the clouds. And of the natural sea spray, it's not easy to sort this out. So we did a bloom indoors, and the green is the phytoplankton, it's the chlorophyll. And what we saw was we got 2 blooms, which was great. And we satisfied both camps. We had basically one case where we saw a huge change in the properties of the aerosols and the cloud properties. And in the other case, no change. And so, that sounds a little strange, but we were able to explain it in that basically it's the bacteria that basically it was the phytoplankton plus the bacteria. The phytoplankton emit or eject sort of all these different big molecules, oily compounds, and we knew that. But the bacteria are quickly trying to digest it and eat it. And so there's this competition between how fast the phytoplankton can emit or produce these organic species that are gooey and oily, and how fast the bacteria can break them down. And so this was the first time that people were actually able to explain this seeming disconnect between how the spray itself and how the seawater composition itself changes clouds. And so it was a pretty exciting time. But I will say, this was the first, 8 years of case. Was just trying to get the biology right and to learn this. And this was a huge, we finally could explain the observations, both sets of observations, which seemed disconnected, and they both are right, but we were able to explain why with this lab and this field in the lab approach. So the last thing I'll just quickly say on case is that we've now, this is what our studies look like on a big scale. Seascape was about I mentioned those gases. So we decided the spray itself wasn't really changing enough. We wanted to know what those gases were doing. And so Seascape was about basically taking that headspace, what comes out over the course of a bloom, and then seeing how what we call not the primary sea spray, but the secondary, the things that form after reactions, Are those what are changing the clouds? And so I won't go into all this. I just have to acknowledge it's a massive team. There were over 90 people that were involved in this study. Two of my former students that are now both doctors, Catherine Mayer and John Sawyer, are, basically were the ones that sort of led this. These campaigns are incredible experiences for students to sort of learn about leadership very quickly. And so I'll just show you quickly what it looks like when it's working, when the whole system is going. It's in one way, it's easier than a field study. And in another way, it's harder because it's got biology. And we found we have to the biology sort of plays the trump card. We have to rely on the biology and hope that it works. And sometimes it does, and sometimes it doesn't. But in the end, it did work in Seascape. And one of the big discoveries we had, which I don't have time to go into, we finally have uncovered that it's those secondary reactions that seem to be really, really important. It's those microbial emissions that we knew about from soil and forests and everywhere. They're also super important from the ocean. I don't know, as I say, in retrospect, I don't know why this is a surprise, but it was a surprise. It has been a surprise. It's now pretty accepted by the community. And now we're working to try and understand what controls those emissions. The ocean's not really thought of as a source of these gases. And so now we're taking this to the next step of the implications for health and for air quality and for climate. So I'll just quickly say, we basically focused on the spray itself and how that affected the clouds. And what we did in Seascape was we basically took things, reacted them, and then looked at the properties of that. And that's sort of where we found the magic answer. So what's next? So basically right now we're building, and I invite anybody who's out in San Diego, you should come see this. This is called SOARS. It's the next frontier. It basically is a huge wind wave channel. Now it's much bigger, as you'll see, than the original one, which always looked big to me. But we can get up to hurricane force winds. We can control the temperatures of the water and the air. We can make sea ice, and we have an integrated chamber where we can do ocean air, reactions. And so now we're just taking it to the next level, and we'll be able to do things like inject CO2 to super high levels, see how that changes the biology, how that changes the emissions. We'll be able to look at feedbacks that nobody else can look at. We can warm the ocean, see how that changes things. So we can do futuristic scenarios, and we can also do past scenarios if we would like. So we're really excited by this. This was also funded by National Science Foundation through the MRI program, and it is just coming online. And as I say, if anybody's out here, this summer will be our first big study on this system. This is what it looks like being built. You can see, this is the other one. I don't know if you can even see the other one. No, here it is. The other one is under here. Remember that long channel? It looked really huge. This is even bigger. And so, basically this is the wind tunnel. This is where the water goes, the water and the waves. And so, this is the system. Basically, we can do sort of all air-sea exchange experiments, bubbles, sort of look at everything. It's an enclosed system, contamination-free. So we can do the same experiments, but now we can add winds and control temperature and many other environmental variables. So we're super excited about SOARS. So I just want to shift gears. One of the things that we've really spent a lot of time on, as I mentioned, is the transfer of microbes to the atmosphere. So this is where my life is shifting over towards microbes. We published a paper in 2018 on this, where we this is this paper by Michaud, where we actually, I should step back. So there was a, paper, it was proposed as early as, the 70s. There was quite a bit of work being done. Blanchard was one of the main people that did this, but they basically said, any place you have breaking waves, you've got bubbles, forming, and they plunge down into the ocean. Those bubbles rise and they scavenge all this hydrophobic material. And so you can end up with these massive enrichment factors of viruses and bacteria over what they are in the water. So you kind of think about bacteria or viruses as being low concentrations. The ocean's huge. They should be dilute. But they actually they can get enriched by 10 to 100,000 relative to salt in the air versus what they were in the water because of this bubble bursting process. It turns out to be incredibly important. And then they turn out to be important. The microbes turn out to be incredibly important. So one of the questions we have is, sort of what about these pathogenic viruses, that are in coastal waters? we have sewage, we have runoff, we have all these viruses going in. So this is what I was doing when the pandemic came along. We were looking at sort of the transfer. So which is like messy, but basically you can see this is bacteria. The red means it was enriched in the air for different types of bacteria. This viruses are down here. And so we looked, and basically you can see there's actually a huge difference between different types of bacteria. Viruses, we didn't see quite as much of a difference, but bacteria we see, certain types get out and certain types do not. It's not the same as just spraying straight sea spray and drying it down. There is something special going on with certain viruses and bacteria. This could be, one way they move around. And people kind of have asked this, do they catch a ride with insects? Do they, how do these things move around the globe? And bubble bursting from water, things that we've traditionally, and this is where Rita Caldwell has been my major hero, things that we've thought about as waterborne, are they just waterborne? The first time I met Rita, she was giving a talk. It was a wonderful talk about cholera. And I went up to her afterwards and I said, how do we know that it's not in the air? And she said, we don't. And so then since then, she and I have been in a conversation and we're getting very close, and I'll talk about it at the end, to doing some work to try and understand this better. So this ties into it. We've been working on trying to understand what happens when you get this massive flooding. We are pumping so many microbes into the ocean. We're changing the ocean microbiome in coastal regions. And so, this is changing our climate, it's changing our air quality, it has to be changing our human health because we're inhaling all this stuff. So we've often thought about, don't go to the beach when the water's polluted, don't surf, but nobody really tells you not to breathe. And so this is the area that we've been studying. This is the Tijuana River, and it has this one area right at the border where there is a wastewater treatment plant that can't keep up with processing. And so millions, sometimes billions of gallons of raw sewage enter our ocean. And so our question is, what happens to it? And so we worked with oceanographers at one point. We put dye into the ocean and, basically released it from this estuary, which is coming along here. And we said, what happens to the dye? Because most people think, okay, you put this much stuff into the ocean is massive. It's just going to escape into the ocean and it's gone forever. Wrong. What happens a lot of times is that it gets trapped in the surf zone. And so we worked with oceanographers who could sort of understand why sometimes it gets trapped. If it gets trapped in the surf zone, that is the recipe for how it gets aerosolized. That is how it can become airborne. If it sails out into the ocean, it's less likely to become airborne. So we, looked at this. Basically, we said, okay, can we see the dye? These are two of my former grad students. Actually, one's just graduating, one is former. And basically, we took these air samplers, we moved them all over San Diego, because once it gets in the air, a lot more people are exposed. It's not just those that live near the beach or walk on the beach. It actually can be miles, can be exposed to this. And so, we basically, sampled the water, the river, the ocean. This is the sewage, actually. This isn't the dye. And we sampled the air to see whether we could actually detect the dye. There's a mimic for if you had pollution. And in some cases we did. So in this case, when it got trapped along the surf zone, we saw it. And in other cases where it went out to sea, we didn't see it. And so now we kind of have proven that, this airborne issue is a concern. We've continued to do studies. I think I have a little bit. Oh yeah, I should mention, we also looked for SARS-CoV-2. So this was my first step into funding from the National Science Foundation to see if we could see SARS-CoV-2. And the answer is, so far, we have found it in the river, we have found it in the ocean, but we have not found it in the air. So that's good news that it's not in the air. The other hopefully good news, although I still would like to better understand it, is, is it infectious? We just detect the RNA, right? We can just say it's there or not, but we can't say whether it's still infectious. There aren't a whole lot of studies on this. People have kind of said, oh, it won't stay infectious. It doesn't like salt water. It doesn't seem to like outdoors. That's all true. But I have to remind you, this is called SARS-2. There was SARS-1, and SARS-1 was from raw sewage becoming airborne, and it did remain infectious. And so I still think I still think we need to do more studies to really understand how the environmental conditions affect how long SARS-CoV-2 remains infectious, but that's another whole talk. So we're moving around now. We're not putting dye in, although we will be next winter. So we've got tremendous interest from the public health officials. You know, we're finding all kinds of things in the air. All the things you find in sewage can get out. And so now the question is, you know, what are the health implications? And we have interest from, as I say, local health officials, the state and the federal government are very interested in sort of this airborne transmission pathway, which has been heavily highlighted through the pandemic. And so we're continuing with these studies and just doing them better and better as we go to learn more and more as we move along. So, you know, one of the things we really want to do is just sort of think about how far these things travel. Do they remain infectious? What happens when people inhale them? So we'll be working with the medical community. This is just a little example. I call this this is like the key, I don't know, the only example I know, a really good example that shows you can link meteorology with an actual disease. This is Kawasaki disease. This is one of my colleagues, Jane Burns, who runs the Kawasaki Disease Research Center. And when the wind pattern's set up properly, so this is wind speed, I believe, in Japan, San Diego, and Hawaii. And when they set up in blue, you can see that there's just this, basically you can just see that the Kawasaki disease cases, Japan, it kind of falls apart, but in San Diego and Hawaii, it's pretty amazing. This is amazing, actually. So she can actually look at the meteorological conditions and decide whether she has to go treat patients or not. So this is, to me, one of the best examples of, sort of, it's hinting at, air, and it's hinting at biology, especially, I should mention, the cases went down in the pandemic with everybody wearing masks and all the interventions that we had. So it hints at biology, but we still don't know what. So we're continuing to work on trying to now identify what the source is that leads to this disease, which is the most common cause of acquired heart disease in children. So it's a really serious issue. I'll just mention there's things we're thinking about now. How do we sample in clouds? How do we sort of look at the microbes that these kids that are infected or that have the symptoms come up with, and how does that compare to what's in the atmosphere? So we have all kinds of ideas on how to do this and, again, do it better. But one of the things I just want to sort of finish this part with, and I'll switch gears in a second. This is my dream project. I've been talking about it. I submitted it as an NSF big idea. And this is the thing that I think Rita Caldwell and I have been, both agree is something that's incredibly exciting, is thinking about, can we create, this is a big picture vision, but can we create a big global map, Google map of, this shows it for all the other aerosols. Why don't we have this for biological aerosols? And so can we do that? And then once we have that, talk about discovery. We can start looking and seeing, how different they are, what controls it day to day, different regions, how that affects the clouds, how that affects weather, how that affects human health. How does it differ in developing and developed countries? There's just so many questions. And with data science being where it is, I think we're at the point where we could really do this. And so I tweeted a few days ago, a tweet that went completely viral. I've got lots of volunteers. If anybody wants to be involved, that's going to turn into a massive, I think, citizen science and regular science project globally. We have volunteers from pretty much every country with just one try. So this is going to be really fun. So stay tuned. I think it's really going to be important as we move forward. So if you look at this map, let me just step back. If you look at this map, this is where I come from. And, this is where I came into SARS-CoV-2 and this pandemic. And I was asked, do you think aerosols go further than 6 feet? And I mentioned, obviously, yes, they go further. So that got me sucked into the pandemic in a pretty major way. So one of the things I did early on, I started writing this in like March and April of 2020. So very early, I started thinking about, what I was hearing, and I realized it had to be airborne. There was a lot of denial going on, and we'll talk about that in a second. And I just wanted it out there. Like, I just wanted to say, it's airborne, and this is what you have to do to protect yourself. And this is why everybody wearing masks, which is shown here, is really, really important, what we call universal masking. So the reason universal masking is important is that the first rule in source control for air pollution or infections, whatever, is you block it at the source. So if it's airborne, you never let it get in the air in the first place. So that's why masks are so important, but they also happen to protect you. They work in both directions. So, the tricky thing that this virus has done is, and the way it's honestly snuck around the globe, is it's being released largely by people who have no symptoms. So people right before they get symptoms, or some people never get symptoms. And so they're just producing these little aerosols that float in the air and don't fall to the ground in 6 feet. And they build up in indoor environments. And so taking a temperature of somebody before they go to an event is honestly pretty useless. And so, it's not coughing and sneezing, as we often think about. It's not touching surfaces. It is in the air. And so, I can't even tell you how many times I've said the words, it is in the air. I've lost track. So why does it matter? Well, this is a historical view, a medical view that has existed, this droplet, we call droplet dogma, that, you produce these big drops that just fall, and that's where all the viruses are. So if you stay 6 feet away, you'll be okay, And the truth is that, large droplets, they do fall, and they have to impact, your eyes or your nose or your mouth, pretty small targets. Aerosols, on the other hand, are inhaled, and they can go deep in the lungs, and they can lead to much more severe disease. So it's really important to think about what is an aerosol and what is a droplet, and how do I control it? Because it's got to be linked. You don't just arbitrarily make it up. So basically, people have been saying if it's above 5 microns, it's going to fall to the ground. In the medical textbooks, that's what it says, and that's not correct. Things up to 100 microns can float all the way across the room. They fall faster, but basically the cutoff should be 100 microns and not 5 microns, which means it's mostly in aerosols, which means it's mostly floating in the air. And so if this had been known, and this, I think, was treated well by a lot of the Asian countries, and they had far less deaths than we did, but we just kept, this crazy science miscommunication is the nicest thing I can say. So early public messaging, February 11th, the head of the WHO, the director, stated the virus was airborne. And then 5 minutes later in his tape, he came back and said, it's not airborne. I didn't mean to say that. And then ever since then, WHO is very, World Health Organization would not go back on this. In fact, they posted a tweet that said it's not airborne, and it's still there. And so this has led to tremendous confusion by the public. We've heard masks work, we've heard masks don't work, we've heard they're on the surfaces, they're not on surfaces. It's just been a disaster in terms of communication. And so a number of us, aerosol scientists, have grouped together. Twitter has become this massive platform for us that we've all gotten connected to people all over the world. Some are aerosol scientists, some are architects, engineers, some are just, people who care and have been following what we say, but we've become we're told we're the go-to source for they trust what we say and how to protect yourself. And so we sort of picked up the slack and have spent a lot of time communicating about this. So this is confusing, right? They say coronavirus, it isn't airborne. We weren't allowed to say that airborne word, but it's definitely borne by air. This is an article back in March of 2020. Can't even tell you how many media interviews we've all done. We did a National Academy workshop in August of 2020. Everyone agreed, all the experts agreed, it is airborne. We did media. This is Lindsay Marr, myself. This is John LaPook, the CBS medical correspondent, who's become a friend of mine. It has been, we've tried every route we can to make a clear message that this virus is in the air. So what do we think right now? Well, right now, finally, we're at the point where we understand there's short range, someone's talking, they're producing these little aerosols that float like smoke, like cigarette smoke. It's like being in the room with a cigarette smoker if you're in a poorly ventilated space, versus a few droplets come out, but not many, and they fall very quickly. And so there's short range and long range aerosol transmission. Basically aerosols float and droplets drop, and aerosols are inhaled. So the evidence, I won't spend too much time on this, but I will just say the evidence is overwhelming. There is no evidence for droplets for surfaces. In fact, CDC has ruled out surfaces. They've said that there's less than a 1 in 10,000 chance of you getting infected by touching a contaminated surface and then getting it into you somehow, right? And so the air is definitely where it's at. And so, what are the common features of these, what we call super spreader events? These are the telltale signs of it being airborne, where one person's sick and 80% of the people that shared that same air all got sick as well. And they didn't lick the same salad fork, right? They shared the same air, and they didn't even have to be within 6 feet, because it will build up over a bigger distance if the air isn't ventilated properly. So, basically, there's all these different examples. There's this restaurant example where the air got trapped at one end of the restaurant, and all the people at these tables that never came near each other, they all caught it. This is one of my favorite ones. This is a chorist that was playing a couple days. They had TV watching to see if they interacted with the people who got infected. His back was actually to those, the ones that got infected are back here. His back was to them. And so again, droplets don't do U-turns is one of my favorite sayings. Droplets just go. They've got so much momentum, they cannot turn around. Aerosols float. And so this again points at aerosols. There's animal studies. Ferrets are a very common animal that's used as for SARS-CoV-2 studies. They put multiple bends. Again, droplets can't make the bends. They've got too much momentum and they just splat out. And so only aerosols could make it. And so they basically took an infected ferret, blew the air, and infected this one, which again had to be aerosols. We published a paper in the Lancet. I'd never published a paper in the Lancet. It was a combination of aerosol scientists and medical folks and science communicators. Basically just said, look, there's at least 10 very solid pieces of the puzzle that they all fit together perfectly. You'd have to explain all of them away to prove that it's not aerosols. We still had people pushing back. We still have people, infection control people, and I'll talk about that in a second. But these super spreaders, the fact there's so much more indoors than outdoors, the fact that surgical masks those little blue masks do not work. Those are designed for droplets. There's just a whole host. And, again, if you're interested, this paper has been very highly circulated and very well received. So why does it matter? Is it just semantics? Am I just making a big deal out of nothing? Why does it matter? And so why do we want them to say that little word airborne? And it's because droplet precautions aren't as good as airborne precautions, and they won't protect people properly. We have lost thousands of healthcare workers worldwide because they have not been given proper protection because of this. And, many people have been given the wrong protection and had the wrong idea and thought they were safe when they weren't because of this little distinction. So it's really, really important. I'll just quickly show that basically, it's a little detailed, but for those who care about the medical side, it's interesting. You often hear about aerosol generating medical procedures, AGMPs. So in those, basically, it's when you're in the healthcare setting, if you're doing one of these, like an intubation or different, they're listed down, they're different acronyms listed down here, but basically they're procedures that cause aerosols to come out, like we thought they did, but nobody measured it really directly before. So somebody actually, this is a great paper by Wilson et al. And what they did was they compared the number of aerosols produced by talking, exercising, shouting, and coughing, just normal respiratory activities. And they compared that to these typical aerosol generating medical procedures. You can see there's hardly any aerosols produced by the most common types compared to just talking. So if you're in the room with someone who's potentially infectious, you're wearing a surgical mask, that's not enough. And so this is the problem. Herein lies the problem with the healthcare workers. Most of the time they're given these little surgical masks that have these gaps. Aerosols just go right out the gaps or right in the gaps. And we give healthcare workers only N95s. Many places will only give them N95s if they're assisting with a known COVID patient and they're doing an aerosol generating procedure. So actually humans are aerosol generating people. I said the P should be people, but this is a big problem. I mean, healthcare workers are still, especially up in Canada, are still being exposed and fighting this. In fact, there was just an article, I don't know if you've seen in the last couple of weeks, talking about going into hospitals and people wearing N95s to be extra safe. And they're being asked at hospitals to remove their N95 and put on a surgical mask. That's how crazy it still is. This is just now finally being addressed. So fit is everything. Filter, fit, and function, as we say. Sometimes people say, but we wore masks and it didn't work. It's not just any mask. Thin little cloth masks aren't going to cut it. Surgical masks, as Delta came and Omicron came and it got more infectious, they don't work. And so, what worked in the beginning doesn't work anymore. And so understanding what masks to wear is super important. So once, again, I was told, do not say airborne. You're going to scare everybody. People are going to think it's the movie Contagion. It's going to be awful, right? And I just said, the public deserves the truth. The public is smart enough to do the right thing. And, that they need to understand why they should be ventilating the air or filtering the air. And so once you acknowledge that it's airborne, it is a totally fixable problem. We wouldn't have had a pandemic if people had just cleaned the air at the beginning. And that was shown pretty clearly in Asia. Basically, because it's been allowed to just go and go and go, because a lot of parts of the world didn't understand, it's now evolved to this horrific situation where we can't seem to stop it. But if we had just never let it get out of control, if we just cleaned the air in the first place, I truly believe we certainly wouldn't be sitting here talking about being in a pandemic still. So where are we now? Well, the WHO finally put airborne. I think they have airborne. Yeah, they have airborne transmission on their site. They quietly put it up on their website on December 23rd, 22nd. They talk about aerosols, they talk about airborne. They just slipped it up there. Still no big announcement, but it's up there. The tweet that says it's not is still up there as well. CDC has come around. I've worked closely, kind of helping them with language. And now they're talking about opening windows and improving ventilation and all the right things. They're telling how we can fix it. The best news, the biggest news is, just a couple of days ago, the White House finally came out with a clear statement. They moved well beyond CDC. And they have basically talked about, let's clear the air. Let's clear the air on COVID. And so this is I just about fell over when I saw this clear, simple statement, the way it's transmitted, airborne particles that can last in the air for minutes to hours. We basically, this is just finally, it's finally out there. So this is really great. They're coming up with a clean air challenge. There's a lot of work going on. They're now telling people how to fix the problem. So one thing I'll quickly say is I am a big advocate for cleaning the indoor air for the same reason people were advocates for cleaning water. We filtered our water long ago. We're not okay with drinking unfiltered water. So why are we okay with breathing unfiltered air? And what I will say is even after COVID, this will pay off. We basically just accept that we always have the flu in the winters. But the flu has gone down with people wearing masks and cleaning the air. And so one of the things, we just published a review on this in Science not too long ago. And it turns out that pretty much every respiratory virus has some airborne component. This one, for whatever reason, is predominantly airborne. The other ones, we're going to have to work out what the sort of relative contributions are. But the point is, there's been a paradigm shift in how we view respiratory viruses. And as I say that, it sounds kind of funny because it's a respiratory virus, and I'm sitting here arguing that it's in the air. Of course it's in the air, but, this is where we are. So one reason, as I said over and over, to acknowledge it's in the air is the public needs to know why, so that they know how to protect themselves. It's that simple. Why should we open the windows and add filters and wear the right mask and, all these things? A big part of it, though, is just protecting our workers, essential workers, all workers. Because if you don't use that airborne, then they're not required to give them the proper protection. So that has been a real problem. And so, we knew this back in 1918. We knew going outside was better. These are kids outdoors back then. And so, we can do things. I I have a little CO2, I don't know if you can see it, my CO2 measurements for ventilation to make sure my air is fresh and wherever I go, I'm holding it here. There's simple ways to make sure your air is clean that you're breathing. The other thing is layers of protection. You know, people will say, what's the one thing I can do? I just say, do as many as you can. It's just each layer adds extra protection as you go. And so you can see this is what's called the Swiss cheese model. You can look it up. It's it's just all these different ways we can actually make sure that we're protected. So I've also helped a number of schools, a lot of schools and businesses reopen. You know, this is a figure made by collaborator Evan Cross, who works, does size measurements. He gives a great analogy. You know, if you don't clean the air, the fish are dying in the pond, and then things get better and better as you clean the air in schools. This is in San Diego Unified, which I helped. We could measure the particles, we could measure the CO2, and we put in the right filtration and ventilation. And we were able to open the schools without major outbreaks for Delta. Omicron turned out to be a heck of a lot more challenging. But for, basically there are ways to do this and to know that you're doing it well. One of my favorite topics, and I think I'm getting close to the end, but one of my favorite topics, I have, a lot of people, you'll hear ventilation, 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 and you'll hear me say filtration, filtration, filtration. And you'll hear me talk about Corsi Rosenthal boxes. I am a big fan of Corsi Rosenthal boxes, and I've become a bigger fan over time because they're so simple. They're elegant solutions. They look kind of clunky, but anyone can build one. And, more people can buy them. Some people are making them and donating them to people who can't afford them. You can build one for about $70, and they outperform a HEPA. HEPAs are great, but HEPAs are slow. HEPAs can't filter because they are so efficient. They don't pass the air that fast. You use these filters like you use in your air systems in your house, your heaters, and you just tape them together with duct tape. These are UCSD's build. And you stick a box fan on top. And so the air gets sucked in through the sides and pumped out through the top. That actually mixes the room air so that you don't, basically you can actually, now ventilation gets easier. They're amazing. And so I started pushing these back in August of 2021. We had a school opening webinar, global webinar, and I just said, with people who've been working with these and testing these, spoke. And then there's just this massive grassroots effort. And I'm convinced thousands of people, thousands of families have been, protected by these filters. In fact, I had people that came to me and others did as well. What do I do? Someone in my family's sick. How do I know the whole family's not going to catch it? Do I just throw up my hands? Is it too bad? Every family, and I'm telling you, there's hundreds of stories like this. Every family that built these Corsi boxes and put them in various spots in their house, nobody else in the family caught it. So, they were also making sure their windows were open and doing other things, but these filters are amazing. So, one thing I'll say is that, the public really needs and deserves clear guidance. It's been a horrible misinformation/disinformation campaign by many people for I don't even know. I guess time will tell why it has been so bad. How many air changes per hour? 6 is kind of the number they give. MERV 13 are the filters. There's pretty set recipe of what puts you in a much safer spot. But the other thing is it tells you what to do, but it also tells you what not to do. And, there's a lot of people that put in these plexiglass barriers. They're terrible because just like this simulation is basically a CFD simulation, you can see that basically it traps the aerosols. These aerosols can also get trapped inside of a shield. So if somebody wearing a face shield with no mask can just have it trapped right there and breathe it in. So they've actually been shown to increase risk of inhalation. So, once you understand how you're getting infected, you can start doing the right things to make sure that you don't. So the last thing I'll say is, are we surprised? I mentioned SARS-1. We shouldn't have been surprised. You look at pictures, there's people wearing masks in 1918. But this really says it all. I remind you that infection is spread through small droplets by speaking and coughing. They knew this back then. This is from 1918. Please abstain from staying in closed spaces, poorly ventilated with a lot of people. All the things that we're now saying that are now people are catching on to, They knew this 100 years ago. So, having said that, I'm going to end there, but I just hope that, by, my sharing with you sort of this crazy saga, that more people can, sort of be made aware, and we can end this pandemic, and prevent future ones. I really think that, cleaning the air is a solution. We should have cleaned it long ago. It's not, we spend 90% of our time indoors. We spend all of our money cleaning outdoor air for some reason. And I will also add that ventilation isn't always possible. With wildfires becoming more prevalent, you can't just open the windows and bring in fresh air. So anyway, I'm going to stop there, and I'm looking forward to a discussion.
Thank you, Kim. That's a bit of an eye opener in many ways. Now for the question and answer session.
I see a hand raised.
And there's a hand raised. That's Carl Merrill. Carl, do you want to unmic, turn on your mic and ask a question?
Yes. First of all, I really enjoyed your presentation. It brought back a lot of memories. I'm a physician, but I do basic research. I was at NIH for 43 years. And I remember, I think way back when I was in medical school, people talked about the epidemics of meningococcal meningitis with the winds in the Sahara Desert and the hot seasons. And so that's been known for some time. And the other thing that your lecture brought to my mind was the argument that Fred Hoyle had made about the fact that there were viruses in the upper atmosphere, and that they were brought down during the sun cycle every 11 years. That's when we'd have the flu epidemic. So I wondered if you'd comment on that. He thought that some of the viruses might be from outer space, from comets. That of course raises a question: has any monitoring been done, say, at our space station for microbes that might be up at that level?
Yes, for the space station, people have swabbed the space station, but it's been mostly, almost all of the airborne has been aimed far more at bacteria. Far less, like we hardly know anything about the virus. There's a couple of groups, as you say, that are looking at airborne, but kind of here and there. But I don't think we know. I don't believe, if it exists, I haven't seen the viruses for the space station. But the focus is mostly for all airborne stuff has been far more on the bacteria than the viruses. But yeah, there's other sort of hoof and mouth. There's other diseases where we've got evidence, that airborne can be really important, but it just hasn't sort of made the general, belief, like it still is. It didn't help us with this one. I will say that. No,
in fact, one of the statements that I've often heard said was that the most infectious virus is measles. And if you have a kid that comes into a room with measles, almost everybody will walk out infected. So I guess that's even more efficient than Omicron. Well,
okay, so that's an interesting point. And it's interesting because why everybody considers measles airborne because of what you just said. And one of the reasons they said this wasn't airborne was because it's not like measles. That's been a hard part. That, to be fair, sometimes I say, I don't know why people, but the truth is that the other weird thing is the Pareto principle for this virus, in that, 20% of the people, 10 to 20% of the people lead to 80% or 90% of the infections. So most of the time people aren't shedding, that much. There's certain, what we call super shedders, super spreaders. And so, yeah, you're right. Like 9 times out of 10, someone can walk in and they're not going to be, it's not going to happen. It's not the right time. There's this narrow window where you're shedding. And if you're it's like Russian roulette, right? If you're with that person at that time, then you see this massive 80% of the people catch it. The thing I also say, though, is it was interesting how the goalposts moved on what it was going to require and why people were saying it couldn't be airborne. And one of them, again, back to measles, was they were saying you can't culture it from the air. Well, measles has never been cultured from the air, ever. This has been cultured from the air now 5 times. So, it's not only been detected in the air, it's been cultured from the air. And so, it's interesting, the comparison, but it's just the fact that it's this, it's like, the R naught you hear about, right? It's, how much it spreads. The majority of people don't spread it. It's the tail that has driven this pandemic. It's the people that spread it to a lot of people. That tail is what, one of the things there's been some really good papers written about. We have to focus on getting rid of that tail, right? We got to figure out how to, cut that back, because that's what's driving it. But that's what's led to a lot of confusion as well.
Thank you.
We have another question from Dick Garwin.
Okay, I found this tremendously exciting and particularly admire the total honesty in regard to SARS-CoV-2 transmission by aerosol and the value of the proper mask and the to and fro of the authorities over the months. Around June 2020, there was a publication that showed a range of at least 1 billion-fold in viral titer and swab from COVID-19 cases. The mode may be 10 to the 4 virions per cubic centimeter of swab material. But by far most of the community virus is in those few people with titers greater than 10 to the 10, 10 to the 7 times, 10 to the 6 times as much. I use a PAPR, a little powered air purifying respirator, so it provides clean air, not in the mask itself, but to the mask. Dollars and works for 4 hours or so, and it doesn't matter what the mask fits a little loosely. Thank you for this wonderful work.
Thank you.
This summer, I hope to see you.
There we go. A question from Jorge Aponte from YouTube. How do CO2 detectors work and prevent COVID-19 and other airborne transmission?
Yeah, so CO2, what it does is it lets you sort of assess. It's the simplest way to assess how, quote, fresh your air is, right? And the freshest air is outdoor air. And we know that the chance of getting outdoors is much lower because it just gets diluted so fast, right? So if you have basically, so a CO2 detector, it's just this little sensor that I showed you. Mine's up to 761 right now. But basically what it does is we know outdoors, thanks to all the CO2 we've produced, it's gone up to, it's about 420 ppm. So ideally, if your indoor air was as fresh as your outdoor air, it would be, about 420. But what happens is if you're in a poorly ventilated space with a lot of people who are talking, when you speak, you produce CO2. And if that CO2, basically if you have multiple people in the room, that CO2, if it's not properly ventilated, will build up. And this can get, 800, 1,000, 2,000, to levels that you're breathing a lot of other people's breath. And so that's when it gets to be really dangerous. In fact, many countries are starting to use this. Japan, you can actually look, like go to the movie theaters, they have the different theaters. You can look at screen 1, 2, 3, 4, and they tell you the CO2 levels in each of the theaters. So, they're putting them in businesses. They're putting them in businesses, and, they're starting to have maps. So if you want to look up your favorite restaurant and see how clean their air is, you can actually look it up on a map, or they're talking about displaying it in windows. But the simple idea is that you're just trying to not have a buildup of other people's breath, which would contain the virus. So CO2 is the absolute simplest way to do that.
Yeah, as Dick Garwin put it in a comment, the CO2 detector tells you what fraction of your inhaled air is someone else's exhaled air.
That's exactly right. Yep, you can calculate it exactly too. Mm-hmm.
So we have a question from via YouTube from Jim Barn. YouTube chat channel. I think you said that since the 1940s in California, quote, we have been seeding clouds, but we have been doing it all wrong, close quote. Is there a correct way to seed clouds?
Like Mother Nature does. If you could figure out a way, and I've had lots of people ask me about this, if you could figure out a way to, get ice at the high levels, just like I showed, that turns out to be an incredibly efficient way to get, the idea is that, every cloud has a certain amount of water that's available to fall. So what you'd like is a mechanism that removes most of that. And the way you do that is by making ice. So if you can make, seed these clouds with some sort of, something that, blankets that area where you're, in our case, it's the Sierras. That's where we sort of store our water. That's our bank supply in California anyway. If you can seed the clouds in those regions in a broad way to create that enhanced process, that would be, that's mimicking the way Mother Nature does it. The way humans do it, and this is for hydroelectric power, it's about 20% of our power, I think somewhere around there in California. So the dams, we try to get enough water to fall where the dams are. So they, the different gas and electric companies go out and they shoot. There's two ways. They either shoot from the ground up into the clouds and hope they make enough more seeds and they make more water fall. It's pretty sketchy, whether it works or it doesn't. They say it gives you maybe 10% more. Nobody really, it's pretty up for debate is a nice way to put it. The other thing that happens, and this happened when we were doing our studies, they have airplanes and they have cloud seeders on their planes and they fly through the clouds and spray out the seeding agents. That the idea being, again, if you make more nuclei, you can get more things to fall and you can get more water to fall. And what I will say is, if you went with the first mechanism, the natural mechanism, what works is making ice at the top, and then it falls and it grabs all the droplets and pulls all that water out too. If you shoot from the ground and you make more seeds, you're going to make a bunch of teeny tiny little droplets. And this is kind of seems subtle, but it's really important. The chance of that scavenging and that efficient removal plummets when you make those tinier droplets. So you're actually counteracting, and reducing the amount of water that will fall if you do happen to be lucky enough to get one of those ice formation events. So you're offsetting, the natural mechanism. It would not work as well if you were, trying to do it from the ground or from an airplane. So I have talked to people, some venture capitalists are interested, sort of, is there a way we could do it? I have some ideas on ways you could do it. There are ways you could think about seeding clouds in a little bit wiser way now that we understand better how Mother Nature does it.
Am I mistaken, or did you say in your talk that there is a certain composition of seeding material that you discovered when you looked at what the composition of the aerosols were, where they arose, that you found that there were certain particular ones that were better at seeding than what people usually use? Yeah.
So as you know, so basically it's pretty well documented. They use it for, protecting crops. They use it for snow machines, right? Bacteria are notorious for giving you. So bacteria, we know certain types of bacteria. And so there's, ones that, again, are well studied and we understand they can make ice at really warm temperatures, as warm as -1 degree C, which is really warm. And dust, different types of minerals do it better than others. So it turns out African dust is a lot of aluminosilicates. And that turns out to be better than something like calcium carbonate, other minerals, I'll just say that, from China. So it turns out certain parts of the world, certain deserts are also better at giving you ice. Again, the answer is you always want it at relatively warm temperatures so that you get more of it. And so, there's different compositions, but the two winners are dust, in a nutshell, dust and bacteria. We don't know about viruses. We're just starting to look at viruses now.
I think it would be a great irony if the best seeding material was from a desert.
It's pretty good. It's pretty good. The African dust was what came across.
Right.
And it's not as good as bacteria, but we're still trying to figure out what the bacteria were, because they probably weren't Pseudomonas syringae, which is the one that everybody uses. They probably, we're trying to figure all that out now. We really need to do a better job at this vertical profiling. We need airplanes, if anybody knows airplanes, companies that can. There are companies that are now starting to put samplers as they fly commercial and collect this stuff. It has such a huge role in our water supply. We really do need to start thinking like that.
So, related question from George Elliott. Can you on mic and ask your question, George? The UCSD grad, by the way.
All right.
Yeah, well, you mentioned that ice is formed at the top, and I'm just wondering how high up is the top? Do airplanes get up that high? or is it above the level that they can fly? And I went back, you sort of answered the question about microbes, but microbes may be used to make artificial snow or man-made snow. Are they used for seeding agents? And wouldn't they be the obvious choice if they are so much more efficient? I mean, you mentioned the temperature at which they could create ice, and it seemed like they would be much more efficient than just about anything else you mentioned.
Yeah, I'm not aware. I can't say it's never been, but it's not common, I will say. The most common seeding agent that's, is shot, into the clouds from these airplanes is silver iodide still. And so, they make them really tiny. They're super tiny, and they can, maybe the idea is they stay suspended. I don't know the motivation why. I agree with you. Like the size, there is a size, the one thing that stands out, and again, I'm just going to throw this out there as conjecture. I don't know that this is the answer, is just there's a big size difference, right? The bacteria are pretty big. So I just don't know. And I do think that there's been people that have been worried, like if you were to sprinkle microbes over land, would that be good or bad for where it's falling, right? In the water and the snow. Do we want that? Well, do we want silver iodide? I don't know, right?
You don't want GMO rain, right? Or you do want GMO rain.
Yeah, that's the question. Right. That's exactly right. I don't know. I'm just throwing it out there. It's a really good question. And it's something I really think we need to do, like we need to think about more, because again, with water, we're seeing its pattern of where it's just moving around, where it's coming down. And I do think we can control it. I do think we can. It's a little weird. Some people say it's modifying Mother Nature. We're modifying Mother Nature right now.
All the time.
It's too late for that one.
That train
But
left
it
the station.
also seems to me that there must be industrial mechanisms for producing microbes in fairly large scale that could then be attenuated, so that some of the worries about dumping bacteria in your rain would be, of course, people are still worried about GMO food. So does it make sense that,
it's one of those, it's one of those things that you think of, like you have to think about the unintended consequences. You're trying to fix one thing and you screw up another, right? But I don't know. I just think that microbes there's a way, I just think there's some really interesting questions, right? That you could think about. There's certain microbes that are really good at taking up CO2. So if they did go in the soil and they help the soil, maybe that would be, I don't know, they're wild ideas, but I like to think about crazy ideas. So these are great questions.
Thank you.
There must be 100 companies that when their first idea, biotech companies, when their first idea didn't work, they turned to producing Bacillus thuringiensis as an insecticide. And that made a lot of this bacteria as an insecticide. I wonder if anybody's looked at whether you could use that to seed. Because it's already being spread all over. It's relatively naturally occurring.
Interesting. We could test that easily. We do ice nucleation measurements in our lab. We measure the efficiency curve for whatever we get. And so that would be interesting. Yeah, we're trying to understand just what the natural ones are, and if there's, how good they are. There's a huge range, I will tell you that. And there are ones that are very good that are coming out of the ocean. I can tell you that. We don't know why yet, but we're trying to figure it out. It's a bit like fishing for the needle in the haystack, because it's 1 in a million make an ice crystal. And so, how do you find it? And I will say we're just going to start using this Bigfoot cell sorter so that we can sort things into the wells and start to get down in lower biomass. We can now sequence lower biomass. So we're drilling down into the details. The questions we can ask are getting better and better because of the technology advances, not for climate, but I'm using them for climate change and health. So
we have a question from Tim Thomas. Tim, you want to unmute your mic and shoot your question?
Sure. Can you hear me?
Yeah, loud and clear.
Good. I was asking about why Omicron is more infectious, in your opinion, or what you think makes it more infectious. Could it be because of transmission through the aerosols? Could it be for people are less symptomatic, so less isolated? Or is it something about it gets infected with fewer particles?
All the above. right? We don't have a way to rule it out, any of those, right? You make me guess. I will tell you that they do know that there were studies, there's one study, so we got a long ways to go, but I think it was with Delta. So, it was shown when went to Delta and it became more infectious that the people were producing a heck of a lot higher viral load, like 1,000 times the viral load, right?
Uh-huh.
And again, no symptoms. And so that's why Delta became so much more infectious. So Omicron, it could be similar. There's all kinds of things that could happen. You could also have, I've been thinking about the production mechanism. It's believe it or not, the production mechanism of viruses on your lungs is very similar to the production mechanism of viruses on the surface of the ocean, believe it or not. And so it turns out that you can also think about things like, the composition depends on where it gets generated. The size depends on where it gets generated. So these are all questions we're starting to think about, like, is it just smaller so it penetrates more deeply, so you don't need as much, right? So there's all these things that can shift. And I just think the answer, honestly, I'm giving you a million reasons because we don't know yet. It's probably some combination. Also, is it just, as you say, is it just more effective somehow at latching on and getting into this, infecting things so you don't need as much? That's also very reasonable. We don't know yet.
Okay, thank you.
Mm-hmm.
Another question from David Rabinowitz. David, you can turn on your mic and ask your question.
Okay, thank you. Apparently, George Eliot's first question got lost in the answer to his second one, so I'd like to raise that again. Is there an optimal altitude for seeding the clouds, actual or pressure altitude?
Yeah. So where it happens, and I don't, again, if we say, let's say Mother Nature has, that's the most enhancement that's ever been shown. 60% increase in snowfall is really a lot. And that happened, let me think, you're making me go back to my thinking of where, I'm trying to remember, it happened in a very specific range of altitudes, where the dust, so the question is, was it special there, or was that just where the dust and bacteria were? And it was between, let me think about this. I want to say, it was between 11 and like 14 kilometers. So that was kind of like the magic, but that could just be where it happened to be, right? As you go up in altitude, obviously it gets colder, And so to me, it just seems like what you need is that combination of, the dust has to be there, the temperatures have to be right to make the ice, right? So the higher the better, because the other key part, which I mentioned, is the higher the better, in the sense that there's all these the clouds, especially when they're convective, are really thick, they're deep. And so the higher the ice forms, the further it falls and the more water it removes. So, I would just say that as high as you can go where the clouds will be interacting and you can actually see would be the key. And we found it kind of it was amazing to discover this, because you can actually use it. They're using it for weather forecast models now, because you can see the dust coming across and you can see the storm coming. And every single time they merge together, drop down and seed the clouds, and you get this enhancement when they're set up. So, but you don't always have the dust there and the microbes, right? They aren't always there. It depends on, the desert they came from, right? But it's kind of cool that they actually are now putting it in weather forecast models. When they see this conversion of events, they know that you're going to get what it leads to. And I didn't say this. I can't believe I didn't say this. It leads to these atmospheric rivers that you hear about. Tremendous amounts of water falling from the sky. And so what happens, what they're doing is they're using it for water resource managers. So if they think the event's going to be really intense, they know to lower the dam levels, so they can collect more of the water more efficiently. So they are using it now. And they do know now dust is everywhere in the atmosphere all the time. And so now they're starting to track that dust and link it with the clouds and the weather as well. So yeah, sorry, that's a long answer but you made me think of all these things. That's kind of like it's almost my past life, but I hope to follow up on it again someday.
Thank you. I really appreciated the full answer, and I really appreciated the talk. Thanks.
Thank you.
Kind of a related question. Do you have a way of studying this empirically? Are there models that you have in the lab where you can do a gradient temperature and pressure. And
Yeah,
and
so there
have,
are models, there's these weather forecast models that are used and they
I mean, in
can,
silico, I mean actual empirical study along the lines of the devices you showed, which are for a different purpose, but this would seem to be something you might actually be able to build a box where you could create something along the lines of what the conditions are where you're trying to seed,
Mhm.
and then put different things in under controlled conditions and see if you can
So
get
we
seeding
do-
to occur.
So we kind of do that. We did this for sea spray. And we didn't do like the whole process, as you say, like there are these cloud chambers that you can, in theory, do this. They're really hard experiments. So there's a lot of disassembled cloud chambers, shall I say. People have built them and tried, and it's hard. It's really hard. It's easier to model it, honestly. But if you just want to understand how efficient something is in the lab, you make like what we call a parameterization that can go into a model, then you can stick it in the model and see how it would change the cloud properties. We have these devices where we actually ramp the temperature and we look at where we put them in the well, like we use a 96 well plate, and we can control the temperature really carefully. We put our samples in there, and then we have it automated now. We can look at which wells pop and how fast for different substances, and we can create what we call an ice nucleation curve, which is different for everything. But that parameterization is then put in climate models to sort of predict the overall effect of that particular, whatever source you're interested in.
That's a really interesting use of 96 well plates.
I know. I can't wait.
I use them a lot, but not for that.
Yeah. So we can't wait to connect that 96 well plate instead of us doing it to the Bigfoot sorter, the cell sorter. So now you'll have a, flow cytometry, optical signals, and you can sort things. Oh, it's going to be, it'll be fun.
I have another question from Jim Van Arstelen. He asks, how has your research to date influenced the design of climate change models? So it's a good follow-on question.
Yeah, it's not as much as I would like. That's where we're going next. I'm talking to one of my colleagues here, Shengping. He's a global climate modeler. That's the hardest model for us to go after because it's just such large scale. When they see me coming with my aerosols of different sources and sizes, they don't really, that's a lot of detail for a climate model, right? So I would say one of the main ways is we have been pushing climate models, again, not as hard, but I'm planning on going back and pushing hard to treat just the ocean correctly. It's not. It's treated as salt. It's treated as, they don't even have any of these other types of really abundant types present. Like they're missing. It's just completely missing. So my goal is, now that we've sort of cased the center that I directed, was really NSF. It was funded by chemistry. So it was to show chemistry is important. And so to climate. But now the next step is to do something with all the measurements we have and to actually improve the models outside, beyond case is what I will say. So we're just now getting to the point where we're putting a lot of our stuff into air quality models now. And then the really, as I say, the really hard one, you asked the really hard one, that's the climate, the overall global climate models. They're just, they have to keep kind of simple. And so the question is, when does chemistry matter? Should we see differences over the ocean, or not, over desert? So we can actually, I think we have enough data where we can start working with modelers. But one of the common responses I get from a lot of modelers, and I'm going to start, as I say, now that I've been in COVID and dealing with all this crazy controversy, I've learned how to sort of get these conversations going. So I'm going to go practice in the climate world now. But warning, here I come. But basically, I mean, one of the things that modelers will tell me, if I say, hey, it doesn't look like sodium chloride, it's not soluble, it doesn't freeze ice at that temperature, like it's all wrong. And I'll say, what if we tell you the right composition and the right ice behavior and the right cloud formation potential? And they're like, oh, my model's not sensitive to that. But that just means they don't have that knob yet. That doesn't mean it's not important, right? So we're in a bit of the measurement world and the modeling world, and we got to fix this. We really got to fix this. We're at a bit of a disconnect, and so I've been talking to more and more modelers and trying to figure out which ones are open-minded to try and kind of open the lid to their engine and start thinking about some of these bigger questions.
A lot of coding. I'm kind
Yeah.
of amused by the idea that you had to prove that chemistry is important, because one of the early PSW meetings, Tyndall came and he talked about the absorptive properties, the greenhouse
Mhm.
properties of various gases, of which he said the strongest was water vapor, which is
Yep.
well known. But he did the other ones too.
Well, the reason the aerosols are so important, first of all, is because the way they're impacting climate the most is through their ability to form clouds. And the models cannot predict where those clouds should be. So that's the first thing we're going to try and do, is see if we can get the low-lying clouds over the oceans correct with the things that we have for the ocean. So that's the first step, because the largest uncertainty in all of climate change is aerosol impacts on clouds. That's number one. The other thing I'll say is, the other way aerosols, so that's called the indirect effect, because it's not just a direct effect with light. The other effect, which is what you're talking about, is sort of the direct effect, how they interact with sunlight. And the reason the chemistry is so hard, it's not like CO2 or methane, in that you just get more warming. Some aerosols warm and some aerosols cool. And the way the models treat that, so sulfate is an example, is one of the types they use in models. It scatters and cools. Soot is black. It absorbs and warms. So what do the modelers use to match the temperature between when their model isn't working very well? They just crank up the sulfate or crank up the soot and get, they kind of use it as a tuning knob. And so now, like, those aren't even real types that we see in the atmosphere. So we've got to work through that. But the other thing I'll say is that, soot, we showed, again, my previous work, we showed that soot mixed with sulfate, it's not separate, they're together. And that is second to CO2 in terms of the overall warming. So, and they're much shorter lived. So if we could figure out the aerosols, we could make changes on much shorter timescales than we can with the greenhouse gases. Not to say we shouldn't also keep going with the greenhouse gases and worry about that, but the aerosols are much shorter term forcers. And so they're things that, if we can figure them out and quit emitting more soot or whatever, we could buy ourselves a lot more time while we grapple with the rest of the problems we face.
I have a question here from Brian Slosky. He asks, he says, I was wondering what sort of particle size distribution you've observed in sea spray aerosols. How broad is that range? And does the particle size of a given droplet have any specific effect on the biology or surface chemistry that it carries?
Those are great questions. We're looking at all that, and they go, let's see. So primary sea spray, not the stuff that forms with reactions that can go all the way, the reactions that make oxidized molecules, and they can grow the teeny tiniest particles. They start at a nanometer. But for primary, the spray itself, it peaks at about 100 by number concentration, peaks at about 130 nanometers. And it goes, or 130, 180 nanometers. So 0.1 to 0.2 microns. And it goes all the way up. You can get droplets up to, 10 microns. Easily, even 100 microns. But we don't talk about those as much because they don't stick around in the atmosphere as long. So we look up to about 10 microns, but they exist. So it's a pretty broad range. I think the smallest size is, I want to say, 50 to 80 nanometers for primary spray. But anyway, so that's kind of the size range. In terms of the droplet size and the composition, there's what matters for reactions is where the surface area is, where the most surface area is. And that is about a micron. And so we look at the reactions that happen on those. So depends on what your question is. If you care about clouds, liquid clouds, you care about the 100 nanometers. If you care about reactions, you care about what's at 1 micron. So it's really complicated, and the composition's different. Sea spray is not the same composition as a function of size because of the way it's produced. So it's incredibly rich from a chemistry perspective. And it's, I will say again, far more complex than just sodium chloride.
So you kind of see a broad range of particle sizes?
Yeah, like several orders of magnitude sometimes, at least 2 orders of magnitude.
And maybe you don't see so much of a stratification of the chemistry, or biology. So one particular larger size has a different biology and chemistry than a smaller size, or?
The smaller sizes where the viruses go for reasons of the bubble bursting mechanism. And the bigger sizes, the bigger droplets tend to contain whole microbes because they're just bigger. So that there's a statistical argument that they will grab a microbe out. So we find bacteria in the bigger sizes, we find viruses in the smaller sizes. Different production mechanisms. One is produced by film drops and one is produced by jet drops. And they sample different parts of the water. So you get very different composition at the different sizes. The break is about a micron. So the smaller stuff has a lot more organic-y goo and viruses, and the bigger stuff is a lot saltier plus some bacteria.
So before we go, I want to ask a couple of more technical questions. How do you sort the sizes? You have a particle sorter?
For our measurements?
I don't understand how you do that because you just have the spray.
For the spray? For like, because
Faucet spray, yeah.
Well, our instrument that I showed you measures it in real time. So we actually track each particle and measure the size of each particle as it's coming in, and then we get its composition by blowing it up. So we take it all as it comes. The speed, right? So the smaller guys move faster, the bigger guys move slower. And so we basically are measuring the size to within 1% very fast. So we measure, it's on the fly, and we can do, as I say, like 50 per second. Depends on how fast our laser can track them. You can go higher than that. So yeah, that's how we do it. We don't, for online. So online, we just get it all at once. Offline, if you want to collect them on filters and go look at them more by electronic process, right? I think that's probably what you're thinking. You typically use what's called a MOODY, micro orifice uniform deposit impactor. And so what it is it's a series of plates, and it separates them. The biggest ones sort of stick on the top stage. Smaller ones make the curve. You get the idea. They flow. And so so basically it's a cascade. The top filter. There's like 8 plates, 8 or 9 stages.
Sure, I get it. Yeah.
And each stage has a range of sizes, and then you slip them out and go do the analysis.
So for typing bacteria, to the extent you're doing it, are you using 16S RNA type of analysis?
Yeah, we're for the bacteria. Yeah. And we're also starting to look more. We've been doing a lot more of like just sort of looking at what the DNA. That's what we've been very focused on. We're starting to do more, obviously, with RNA as well. So we're starting to use different sequencing systems just to get lower. We're really trying to push this lower biomass question because you need it for the air.
How are you looking at viruses? Is it just doing shotgun sequencing, or the sequences that you look for, or?
So for the viruses, we've been just doing so far we haven't done any of the. We will do shotgun. The idea is to do shotgun, but for the SARS-CoV-2 work we've just been doing qPCR, and so.
Okay, so you know what you're looking for. So that's a whole different ball of wax.
Yep.
I just wondered. And what you said about making these CR boxes, you use MERV 13 filters, and they actually work well enough to remove these particles?
Yeah, so there was some confusion early on. People thought like a HEPA. It's like, the HEPA basically removes 99.995 or some ridiculously high number of particles in one pass. And so people initially with the boxes thought, okay, we need a MERV 15, 16, the higher the number, the higher the efficiency. Turns out it's better to run with a little bit lower efficiency, because then it pumps the air through much faster. And so you get much higher, what we call, it's called clean air delivery rate. I never thought I'd be talking about this, but anyway, it just makes it just cleans the room really fast. You can drop the level of aerosols, smoke, pollution, allergens. I sound like I'm selling them. I don't. I have no conflict of interest. But you can drop the levels with one of these Corsi boxes in a room in about 10 minutes. 90% of them are gone, just gone. And everybody who has them, that's built them, tells me they love, like their allergies have gone down. Their family's not all sick. They said that, they have pets and they don't see stuff floating in the lights anymore. Air is clean. So they're pretty awesome.
Maybe you should go into the business.
I know, maybe I should. My son sometimes asks about it. Mom, don't you think we should? Because people want them. Like some people don't want to be. And one thing people say, oh, how could I build it? It's got to be hard. Literally, you tape 4 filters together and stick a thing on top, right?
Yeah.
And so some people, the guy Rosenthal, who's done a lot of the work, he's been awesome. Jim Rosenthal. He describes it as if you can wrap a package to mail it, you can build a Corsi box. So anyway. They're pretty simple.
Interesting. Be astonishing if that turns out to be a real solution to our current.
Yeah, I mean, I think we can get a little more elegant with them. I mean, but they work. I've been thinking about, okay, could you make them into like a coffee table? Some people have made them into look like an amplifier, in a band. They've made them look. They've tried to blend them in. They've used really cool tape and kind of made them artistic. There's all kinds of designs.
It's a cube. There's lots of things you can do with cubes.
Sorry.
It's a cube.
Yeah. So
a lot of things you can do. Anyway, Thank you so much. It's been terrific. It's a real eye-opening in many ways, and I'm very glad that you shared your time with us. I'm sorry that you're not here in person, but as you know, you have a rain check to come and join us whenever you're in DC. Hopefully when somebody else is giving a talk, so you can have a full PSW dinner. But if not, we will arrange something for you. Maybe when you come to DC for your National Academy of Sciences induction, although I imagine you're going to have a few other parties to go to.
It's going to be, I can't wait to come back to, I love DC. So yeah, I'll definitely let you know. Thank you. Thank you so much. I look forward to it.
So before everybody goes, we have a few closing announcements. We're planning for the next meeting to be in person. Of course, that will depend on COVID and the COVID-related measures in place at the time, but it's looking pretty good. Our next meeting will be on April 8th, our 2,457th meeting, and the speaker will be Shabita Satyapal, professor of physics and astronomy at George Mason University, and she will be speaking about supermassive black holes. The 2,458th meeting will take place on April 22nd. Our speaker will be Elizabeth Strychalski. She is the group leader of the Cellular Engineering Group at the National Institutes of Standards and Technology. And she'll be speaking about living measurement systems made from cells and on the minimum gene set required to make a normally functioning cell. The speaker on May 6th at the 2,459th meeting will be Rahul Saini. He is CEO of Teleatry, MEMS director at NanoRetina, director of micro and biosystems at Zyvex Laboratory, and CTO at NXT STEM. He will be speaking about implantable electronic devices that are set to revolutionize certain aspects of medical care, illustrated by an implantable retina, now in clinical trials, and an implantable vagus nerve stimulator that can be used to control epileptic seizures. The 2,460th meeting will be held on May 20th. Will be our Joseph Henry lecture, I hope. The speaker will be Bob Belaram, who is at JPL Caltech. He is the chief engineer of the ingenious Ingenuity helicopter that is currently making flights on Mars. And he will be speaking on Ingenuity, the first flying machine on Mars. The 2,461st meeting will be on June 3rd. The speaker will be Charles Tahan. He is the head of quantum computing at the White House Office of Science and Technology Policy and chief scientist at the Laboratory of Physical Sciences. He will be speaking about quantum computing and the National Quantum Computing Initiative, which he heads. The speaker on June 17th at our 2,462nd meeting will be announced at a later date. Please check the PSW website often for up-to-date information on meetings and whether lectures will be in person or via Zoom, which in future we will try to post prominently. COVID and COVID-related public health measures are constantly changing, and PSW must adjust its plans accordingly. We hope you will join us for these meetings, whether they are in person or conducted like tonight's meeting via Zoom. Finally, before we go, let's thank tonight's volunteers for producing this meeting. Robin Taylor, our Zoom director. James Heelan, our recording secretary, for reading the minutes, preparing and reading the minutes. Cameo Ann McQueen for handling the YouTube chat. Thanks to all of our volunteers who put an awful lot of time and effort into these meetings. We greatly appreciate it. PSW wouldn't function without them. And with that, I will now adjourn the 2456th meeting of the Society. I wish everyone a good morning, afternoon, evening, or night, wherever you may be, and have a good weekend. The meeting is adjourned.